Hi everyone, my name is Melina Mathur and I am the Product Manager for Biopharma here at Twist Bioscience. It's my privilege today to share with you how Twist Biopharma's offerings can help you expedite your cancer drug discovery. In 2019, 6 of the top 10 best-selling drugs were cancer treatments, with 3 of them being monoclonal antibodies. Monoclonal antibodies used in cancer treatment are designed to target certain antigens on the surface of cancer cells. Over the past 20 years, antibodies have emerged as important therapeutics for cancer that have transformed patient care. More than a dozen monoclonal antibodies have been approved by the FDA to fight different types of cancer, including breast, lung, and melanoma. And monoclonal antibodies have extended some patients' survival by as much as 10 years. Despite their therapeutic potential, development timelines can be long, and the average time to approval for antibody therapies for cancer is about 8 to 9 years. Here at Twist, we would like to help you expedite your cancer drug discovery process. And in today's presentation, I'll share with you Twist's offerings to help you in your journey towards developing the next big cancer therapeutic. At Twist, we streamline antibody development by speeding up 2 time-consuming steps in the drug development process: antibody discovery and antibody optimization. In discovery, we discover an antibody that binds and modulates the function of the target of interest. Here, we use our focused DNA synthesis to construct phage display libraries and enrich them for high-affinity clones. In optimization, we optimize your existing lead antibody for greater potency and developability. We can modify lead candidates to ensure favorable characteristics such as humanization, altered glycosylation, etc. Even if your antibody was derived using a different platform, you can bring it into the Twist platform and use the Twist platform for your optimization purposes. So in today's talk, I will be highlighting 3 key products and services that we offer to help streamline your drug discovery process. First, we offer our Library of Libraries, which is a collection of synthetic antibody libraries. Second, we offer our Antibody Discovery service. And third, we offer our antibody optimization service called TAO. I'll dive into these offerings next. First, we have our Library of Libraries. In the past, a lot of discovery companies were phage display companies that would have one single library, and they would use that library over and over and over again. Partners would be limited by the overall breadth of that library and also by the overall diversity of that library. Here at Twist, our solution is to develop a new library of libraries, where we can quickly make a set of high-quality libraries using different strategies, each of which contain over 10 billion different antibody sequences in them. By creating this Library of Libraries asset, we give you greater breadth in the number of antibodies that you can screen against your target. So you might be wondering, how can we quickly make so many different libraries? Our Library of Libraries really sits upon our key competitive advantage, and that is Twist Bioscience's DNA synthesis technology. This synthesis technology provides game-changing throughput and cost through quality and speed at scale. Through this approach, we are able to develop synthetic antibody libraries that have exact base-by-base precision as we create these using oligopools and then encode them into our libraries. We have strict control of our codons that we use, and we also have fantastic combinatorial diversity by mixing different oligopools as we build our libraries. And we have tight control over the amino acid distribution as we build them. Putting in length distribution, particularly for heavy chain CDR3s, is easy because we can just make oligos of different lengths. This is in contrast to current present-day technologies such as degenerate oligos, where you would have to generate a new set of degenerate oligos for every length that you were interested in. And because we're making each sequence explicitly, we can also remove restriction sites and unwanted motifs or sequences that might lead to developability risk. Oftentimes, we encode for multiple germlines as we're building our libraries, and then at the end, we very importantly do QC and NGS on all of our libraries to really validate the quality and make sure that what we're offering is top-notch. So using this technology and our goal to build out this library of libraries, we have already built out a comprehensive set of over 15 general and target-focused libraries. These include the libraries in this top row here, which are our general use, and the ones in our bottom row here, which are target-focused. And these really enable antibody discoveries on many fronts. Focusing in on the top left, the hyperimmune libraries, we have our library series that we call our hyperimmune libraries, which is fully human Fab library that uses natural human CDR3 diversity. We've actually created an oligo pool of over 2 million heavy chain CDR3s that we encoded to this library. We've also made a common light chain version of the same library that we can use for making bispecifics. Next, we have our VHH libraries. This series is of VHH single domain libraries. We started with a very large database of llama sequences that have discrete antigen specificity, and then we created 4 different libraries with different design schemes, either based on using ratios of amino acids or just shuffling natural CDRs. We have our structural scFv library, which is built using a database of all known antibodies that have ever been crystallized. Our ancestral library is a new synthetic antibody library developed using trends observed in a curated yet broad set of therapeutic antibodies. Our Minotaur library is a new synthetic antibody library that grafts ultra-long HCDR3 loops from bovine antibodies into a fully humanized antibody framework. And our AI hypermutated library uses a neural network which mimics B-cell receptor recombination and hypermutation and produces antibodies with developability in mind. When focusing in on our target-focused libraries, we have libraries that are looking to develop antibodies for GPCRs and ion channels and carbohydrates. All of these are hard-to-drug targets that have historically been very difficult to identify antibodies against. So we offer really exciting and very robust antibodies in this space to help you develop antibody therapeutics building off of this library of libraries. You can choose to either license a library itself, or you can use one of our libraries to form the foundation of an antibody discovery project with us. With these antibody discovery partnerships, we offer speed and automation. These projects take a few months. And in terms of the overall workflow, because we have these libraries ready to go off the shelf and they're high quality, we can go ahead and pan them against your target of interest and screen them. We can even generate custom libraries for you if you would like to go through our antibody discovery process using a custom design library. We do panning with phage display that is either cell-based or bead-based, depending on whether your target is soluble or is a cell surface target. And we follow up and do NGS and Sanger sequencing for about a week. We don't mess around with antibody fragments, and because we sit in a DNA synthesis company, we can very quickly reformat the sequences and express them and purify them, and then take them through a whole series of binding and functional assays. As our partner, all you need to do is provide us with your target of interest, and we'll do the rest. As a quick example of how we've been able to really leverage and use our antibody discovery process, we have been able to identify a potent antibody antagonist against A2A, an adenosine receptor. And this receptor blockade represents the potential for next-generation immune checkpoint inhibition in cancer immunotherapy. Here we show that this antibody lead is a high-affinity binder to the human A2A receptor and is a functional antagonist in vitro and in primary T cells. And finally, we showed that this is a strong lead candidate in a cross-reactivity study where our lead binds just its cognate receptor. This example really highlights how quickly we can identify high-quality antibody leads against meaningful targets in oncology. Finally, we have our Twist antibody optimization platform. The idea with Tau is that you're taking an existing antibody that you already have some sort of affinity against and improving it so that you can take it forward with improved characteristics, whether that's better binding, or you might want to bind multiple of the same antigen of different species, or you may want to narrow that and only bind one specific isotype. The nice thing about Tau is that you can basically take any antibody, a non-human antibody or a human antibody, and then we find the closest human germline that it's associated with and pick that as its framework. We humanize the antibody and also try to improve its binding affinity at the same time. Tau is great because it offers the ability to create optimized Tau antibody candidates that follow the rules of the natural human repertoire. A typical Tau project with us takes about 4 months, and all you need to do is to provide us your antibody sequence. We input that into our software, generate an optimized library, and then screen against that library. As a proof of concept example here, we have shown that we're able to use Tau to optimize an important target in cancer, PD-1. we were able to increase affinity by 72 times using our Tau platform. As we've developed the products and services I've shared with you, we've also assembled a pipeline of preclinical-ready monoclonal antibodies. These range from ADORA2A, PD-1, TIGIT, CD3, and CXCR4 in the cancer space. If you're interested in engaging in licensing discussions, please reach out to us. In conclusion, there are multiple different ways that we can help you achieve your goals and different ways you can work with us here at Twist Biopharma. You can license our incredible library of libraries. You can partner around generating new leads against any target of interest. You can also partner with us around optimizing an existing antibody lead that you're excited about. And finally, you can also work with us around leads that we have already generated some proof of concept around in case you're interested in licensing them. In addition to the products and services I highlighted today, you can also engage with Twist Biopharma for custom library generation or for high-throughput IgG production. To work with us, and if you have any questions, please email us at biopharmatwistbioscience.com and we'd be happy to help you. Thank you.